Hello everyone. In this video we want to continue talking about the cell cycle. We finished last time from the interphase and how at the end of the interphase the cell prepared itself to copy the DNA to be ready to the next phase, which is the mitosis. For the mitosis, actually it happens in eukaryotic cells, so you have to know here for mitosis here is a part of the cell cycle during which the nucleus divide. You have to notice here, which is very important to know here, the cell here includes the nucleus divide. Okay, you have to put in your mind for mitosis it doesn't occur in prokaryotic cells. As you know, the prokaryotic they don't have a nucleus, so that's why no mitosis. Okay, so mitosis result in two. What is the result after the end of the mitosis? The result two identical cells. They have the same genetic material. Okay, so here the same genetic material. So during mitosis the chromosomes then condense to form from the chromatin. Actually for the interphase we said the DNA start to be duplicated, and here it start to be condensed, and it will make like the X-shape structure, which is the chromosome. Okay, so if you here, as you see here, if you look under the microscope at any cell during the mitosis, you will see the chromosomes will be visible because they start to be condensed. Okay, so they will be it's easy to see these chromosomes. Okay, so at the end of the mitosis, the cell has two identical set of chromosomes in two separate nuclei. Listen carefully to this video to make sure that you understand.
Mitosis has four main stages: prophase, metaphase, anaphase, and telophase.
Okay, so mitosis here, when the cells start to divide, we will talk about these phases in more detail. Just listen now. We will explain these phases. All of these phases, I'll explain it for you.
Prophase is the first stage. During prophase, chromosomes condense. That means they get shorter and more compact. Also, the nuclear membrane breaks down.
Let's stop. Actually, for mitosis is not only one step. For the whole cell cycle, three stages, right? Three phases. But for the here in interphase, no cell division. But in mitosis, where the cells start to divide, there are how many phases? There are four phases. Okay, let's talk about the first one now. For the first one, which is prophase, what happened during the prophase? During the prophase, the DNA start to condense. It's gonna be like very condensed, and it's gonna be like shorter. Yani imagine it's like this. Just hold on. Condense and shorter. They will be visible under the microscope. Okay, so chromatin in the nucleus of the cell condense and becomes visible under a microscope. They start to make this X shape, and they start to make as chromosomes. Okay, here each chromosome consists, as you see, of two chromatin, right? Chromatid, sorry, and they are held by what? They are held by centromere. Here the other thing that happened in the prophase, the nuclear membrane that surrounded the DNA, the genetic material, start to breaks down. It will be removed. The DNA here, look at this cell here. The first thing, the chromosomes here start to be condensed. Chromatin start to be condensed and make the X shape, and also start to be condensed. Also, the nuclear membrane, it will be be removed. It will be removed from it will be breakdown. Okay, so here, if I want to talk about the phases, let's talk about mitosis. for prophase, which is the first one. And the prophase, look at this one here, the chromatin and the nucleus condense in the into chromosomes. They will be like X shape. The membrane around the nucleus breaks down. So here write down what are the things that happen in the first stage of mitosis, which is what? Which is a prophase. The first one, condense into chromosome. 1. 2, the membrane around the nucleus, it breaks down. Okay, let's continue now talking about the mitosis processes. After prophase, what happened? Listen carefully.
The chromosomes couldn't move where they need to go if the nuclear membrane stayed intact, could they? The next stage is metaphase. During metaphase, the duplicated chromosomes line up in the center of the cell.
Okay, so the chromosomes now are formed, right? And they condense. In the second stage or the second phase, which is metaphase, in the metaphase, the chromosomes, they will line up in the middle or in the center of the cell, will be here. Look how they are lined up here in the center of the cell. Continue.
One member of each duplicated chromosome pair faces toward one side of the cell, and the other faces toward the opposite side.
Okay, so here if you look, these are the chromosomes 1, 2, 3, 4 as you see them, right? For this one, this is the first part, which is the chromatid 1, and this is the second chromatid. Yani each chromatid to each side of what? Of this cell. Okay, so and I want to add here something they didn't mention. For metaphase, the chromosomes line up in the middle of the cell. Centromeres of chromosomes are the same distance from each side of the cell. Okay, and as you see here, like these things here, and they have like fibers. Like, can you see these fibers? Now, what are these? Just hold on to show you. These fibers are called mitotic spindle. These mitotic spindle, they start from each side. These are centrosome. These are called what? Centrosome. Look at them. I'm going to write the name of these centrosome. And the fibers are mitotic spindle. So let's write the centrosome first. Centrosome. Okay, and just hold on. And these fibers are called what? I'm going to write them here. The fibers are spindle, mitotic spindle. What are these? Actually, the centrosome start to produce these fibers, this mitotic spindle. Okay, and each one from each side, this spindle, it will attack to the first chromatid from this side, and the spindle mitotic spindle, it will start to attach with the chromatid from the next side. Yani the chromatid number 1 here, these are they are a sister chromatid, as you see, right? The chromatid number 1, okay, will be attached with this fiber. I'll call it fiber number 1, and chromatid number 2 with 2. The same thing for the next fiber. It will attach this one with this chromatid, and this one with this chromatid, okay? And this one here start to attach with this chromatid, and this one with this chromatid. Actually, what happened, or why these are happened here? Let's move to the next stage to understand why they are attached. So in the metaphase, two things are happened. The first one, the chromosomes line up in the center of the cell, and the mitotic spindle start to attach from one chromatid from each side. And this mitotic spindle will attach with the first chromatid, and this mitotic spindle attach with this chromatid, and so on. Let's continue.
Phase is the third stage. During anaphase, the duplicated chromosomes separate and move to opposite sides of the cell.
Okay, let's stop here. At the beginning, prophase, what happened? The chromatin in the nucleus of the cell condense and become visible under a microscope. They will become like X shape, okay? And as you see, and also the membrane around the nucleus, it will be removed, okay, broken down. Here the centriole or the centrosome, they are here actually, okay? They start to go to the opposite side of the cell in the metaphase, and the chromosomes will line up in the middle of the cell, as you see, and here the mitotic spindle start to attach with each chromatid for opposite side, as you see. Then in phase number three, as you see here, these mitotic spindle or these spindle fibers start to be pulled to the other side line. Look at this one. They pull the chromatid and they become shorter, as you see, and they start to separate these sister chromatid from each other. And as you see, they are separated now. This one, it was attached with this one in the centromere, but now when the mitotic spindle becomes shorter, they will pull them to the opposite side. So in anaphase, the chromatids separate. They are pulled to opposite side of the cell. Again, they are pulled to the opposite side of the cell. Each side of the cell ends up with a complete set of a chromosome. Now, these will be a complete set of a chromosome, and this side will be complete set of a chromosome. Okay, let's go back here. This is the prophase we finished. You see how these chromosomes line up in the metaphase and how they are attached with the spindle fibers or mitotic spindle. Now in the last, in this third stage, which is anaphase, they will be separated to the opposite side, okay, and how the mitotic spindles start to be shorter to pull them from each other, to separate them from each other. Now in the last stage, listen carefully, or before actually, which is the telophase. Listen to the telophase.
So that each side gets equal numbers and types of chromosomes. During the final stage, which is called telophase, nuclear membranes form around each set of chromosomes.
Okay. Now, during the final stage, which is telophase, what's gonna happen? The nuclear membrane start to be formed. Now these sets of chromosome in this side and this side, this one start to make a nuclear membrane around it, and this one, it has another nuclear membrane around it, and start to be around each set of chromosomes, as you see. Look how it start to be pinch inward. Now the cell start to pinch inward. Look how it pinch inward like this. Okay, it's like a balloon or a dough when you want to cut it. Okay, look now at this one. So here, if I want to write the notes for the telophase, for the telophase, a new nuclear membrane forms around each group of chromosomes, which become less condensed. The chromosomes, they will become less condensed, not like when we started in the prophase. Okay, now what about cytokinesis? Also, cytokinesis is started, which is the third stage of the cell cycle. It started in the telophase. What happened in the cytokinesis? Here in the cytokinesis, listen carefully.
After mitosis separates the duplicated chromosomes into two nuclei, the process of cytokinesis separates the rest of the original cell into two.
So here is the telophase, a nuclear membrane are separated. They are formed. The new nuclei are formed. They are separated from each other. The cytokinesis, actually, what is the cytokinesis? Is the division of the parent cell cytoplasm. So cytokinesis begins during the last step of mitosis, okay, which is in telophase. Okay, so during cytokinesis, the cell membrane bends inward, as you see, between the new nuclei. Look at this one here. Look how it starts to pinch inward. Okay, eventually, and the pinch all the way forming two complete cells. In a cell that has a cell wall, for example, if you remember for the plant cell, we said they don't pinch inward, they are making what? The cell plate. Actually, for the cell plate becomes a cell membrane that separates the new cells. A new cell wall form where the plate was. Okay, so you understand now these stages for mitosis. Mitosis it happen actually for the somatic cells. The somatic cells, the body cells, to make a new cells. These cells, they have the same number of the genetic material. Okay, guys. Next time I'm going to make like another review for these stages of mitosis. If you have any question about the cell cycle, you can send me your question. Thank you for listening.